Hello, my name is Rory Boyle. I am a postdoc in the Harvard Aging Brain Study in Massachusetts General Hospital and Harvard Medical School. I'm co-presenting today with Yihe Weng, who is at the Whelan Lab in Trinity College Dublin. Yihe will present this live on today at OHBM, and we are presenting a method for connectome-based predictive modeling for large neuroimaging datasets. So connectome-based predictive modeling, or CPM, is a data-driven modeling approach that enables the prediction of cognitive or behavioral phenotypes from functional connectivity data. So very simply, the method takes fMRI scans or functional connectivity data in the form of connectivity matrices from fMRI scans and relates this connectivity to a phenotype of interest. It produces three summary measures: positive network strength prediction, predictive values, negative network strength predictive values, and combined network strength predictive values that reflect the connectivity strength of connections in different networks that are related to that phenotype. These connections can then be further studied using visualization techniques like glass brain, circle plots, or heat maps for different internetwork connectivity visualizations, or also different computational methods, which we'll touch on further. So CPM came to prominence in 2015 following a publication by Emily Finn and colleagues from Todd Constable's group in Yale, and where CPM was used to predict fluid intelligence from resting state functional connectivity. Following that, in 2016, Monica Rosenberg and colleagues from the same lab applied CPM to predict sustained attention from task-based functional connectivity data. And then the next year, in 2017, Shen and colleagues published a protocol paper in Nature Protocols, which provided detailed instructions and code for running CPM on users' data. So this has led to considerable yearly growth in the number of publications applying CPM. And when you search connectome-based predictive modeling in Google, you get over 1,500 results. So it's quite widely used now at the moment. And there's various number of reasons for this growth in use. Number one being the interpretability of the model. So it provides these three single value summary measures that summarize complex data from connectivity matrices into single scalar values. And these reflect connectivity strength across relevant connections that are significantly associated with your phenotype. And so positive network strength refers to the correlation strength or connectivity strength of connections where higher correlations are related to higher values of the phenotype. And negative network strength reflects connectivity strength in connections where higher connectivity values or higher correlations are negatively associated with the phenotype. And combined network strength simply is an overall measure of connectivity strength across all connections in the brain that are associated with that phenotype. So I suppose the ultimate or desired use case for CPM may be to use these measures to track changes in the phenotype across time using fMRI data. Another reason that it's widely used is a data-driven approach that uses whole brain functional connectivity data. So the modeling approach is not constrained by a priori chosen regions of interest or networks. And this is important because there's evidence that individual differences in cognition are better predicted by whole brain data rather than specific ROI or localized data when using fMRI. So that's from a cerebral cortex paper in 2021 by Zhao and colleagues. So this may lead to improved predictive accuracy when using CPM. CPM also enables really nice and relatively easy neurobiological insights. So you have Bioimage Suite, which is a free online web page or web application that takes CPM sort of output and produces some really nice visualizations like these circle plots and glass brains and network heat maps. And then there's also additional computational methods that can further allow you to probe the relevance of specific networks or connections or regions to different phenotypes. So this one method for one such computational method is computational lesioning, which is where you apply simulated or computational lesions to the connectivity matrix before running CPM. And these lesions simply involve removing nodes pertaining to specific connections or within certain regions of interest or networks. And you then compare the predictive accuracy of the lesion matrix or lesion data to the whole brain data to evaluate whether that region of interest or network was vital for accurate prediction of that phenotype. So in 2021, in a 2021 cerebral cortex paper, Weng and colleagues demonstrated this by comparing 3 lesion predictions. So one with a lesion to the limbic cortex, one without nodes in the prefrontal cortex, and one without nodes in the temporal lobe. And they demonstrated that computation or simulated lesions to the limbic cortex or temporal lobe generated significantly less accurate predictions of anxiety compared to whole brain data in green. So this suggests that nodes within these regions are highly relevant to the phenotype of anxiety. Another reason why it's widely used is that various studies have demonstrated external validity or generalizability of CPM measures to independent data sets. And interestingly, it's also been shown that CPM models may generalize across modalities, so from task state to resting state fMRI data. So this was demonstrated by Monica Rosenberg and colleagues in 2016, where they developed a measure of sustained attention using task-based functional connectivity to predict a sustained attention phenotype. And then they applied this to resting state functional connectivity data in an independent data set, and they were able to predict attention. So potentially this could mean that a measure could be developed in one large data set and then shared with researchers for use on their own smaller data sets or even data sets of a different modality in some instances. So connectome-based predictive modeling has been widely applied to model various cognitive and behavioral phenotypes, as you see here from a list of different citations. If anyone would like to access these or get these actual full references, please let me know and I can email them on. But yeah, it's been widely applied, and that I suppose shows the popularity of the technique. So today, we'll provide a demonstration of a flexible CPM method, which we have optimized for large neuroimaging data sets. So our method enables fast and efficient analysis of neuroimaging of large data sets via the use of parallel processing. And then given that many large neuroimaging data sets are comprised of data from multi-site consortia, our method enables researchers to account for site and scanner effects in these data. So you can account for these effects by controlling for site and or scanner type as a covariate in the CPM analysis. We also provide an additional least site outcross validation methods where you can train the model on, say, n-1 sites and then test your model on the left out site. And while not strictly relevant to large data sets, our method also provides some further flexibility for running CPM because it enables permutation testing, it provides for applying computational lesions, and it allows for different cross-validation schemes and different methods for feature selection or edge selection. So typically the input data or the functional connectivity data for CPM papers follow standard pre-processing pipelines. So some papers have used the Sirich, the 36 parameter pipeline outlined by Sirich and colleagues in 2017, whereby you regress out the motion parameters as well as the mean white matter, mean CSF, and global signal. Their derivatives, the quadratics and squares of derivatives, you regress those out of the data. Other sets of parameters have also been regressed out, so 24 parameters where you don't regress out global signal. However, global signal regression, while obviously debated quite a lot, is recommended here, as Abigail Green and colleagues have shown in 2018, showed that global signal regression does improve the predictive accuracy of CPM. Various different smoothing and filtering methods have been used, but most often the most often used method is temporal smoothing with a zero mean univariate Gaussian filter. And then by far and away the most common method for parcellation when using the CPM is to use the 268 node Shen atlas, which is shown here, along with the different networks that these nodes are, which they form part of. So the data are parsed into this, and then you basically use 268 by 268 connectivity matrices as the input to the CPM. So before we demonstrate the actual example today, I'd like to take you through the basic method step by step to ensure that it's understood. So the first couple of steps when running the CPM are to split your data into your train and test folds. So for a simple explanation, we will explain leave one out cross-validation here. So this is where, for example, if you have 5 participants, you leave one aside and you fit your model on the 4 retained participants and then apply that model to the left out participant at the end. You then repeat that for each remaining participant in your data. So you'd have 5 iterations or 5 attempts at this. So in step 1, you leave your test participant out, so leave them to the side, and then you focus on your training set or your training data. So in the training data, you correlate each edge in the connectivity matrix across participants with your target variable. And following the correlation, you then obtain the statistically significant edges. So beyond some p-value threshold, typically the p-value threshold is 0.01. And you separate these edges into edges that are positively associated with the phenotype and edges where correlation strength or connectivity strength is negatively associated with the phenotype. And then in step 4, you sum those significant edges. You sum the positive edges to form your positive network strength value, and you sum the negative edges to form your negative network strength value. So again, negative network strength simply refers to the connectivity strength, the summed connectivity strength of connections where connectivity is positively associated with your phenotype, and negative network strength is where the sum is equal to the summed connectivity strength of connections that are negatively associated with your phenotype. You can then further calculate combined network strength, which is termed here in the figure combo network strength for brevity, and by subtracting your negative network strength value from your positive network strength value. And again, combined network strength refers to, it's a summary measure of connectivity strength across all connections that are associated with the phenotype. You then regress these 3 network strength values on your target variable, and you obtain your fitted model parameters. So you do this separately using a linear regression for each of your 3 network strength values, and you obtain the model parameters, which are a model intercept and a model slope. And you save these and store these for use later. You then move back to your test set participant. And using the edges that were significant for your training set, you calculate again your network strength values for the test set participant. You then apply these fitted model parameters. You then apply the fitted model parameters for the linear regressions to these calculated network strength values to generate predicted values for your test set participant. So you'll have 3 predictions, which would be the prediction based on your positive network strength, prediction based on your negative network strength, and prediction based on your combined network strength. So your positive network strength prediction, negative network strength prediction, and combined network strength prediction. Again, as we're using a leave one out cross-validation framework, you then move on and leave out your next participant, and you go back and you repeat steps 2 to 7 for that left out participant to predict the target variable for that participant. And you do that for every participant. It's such that every participant in your data set gets obtains your 3 predicted values, or you get a prediction for each of your participants in the data set. So again, before I demonstrate the example, there's one more step I'd like to do. I'd like to discuss the different decision points that arise when applying our flexible CPM method. So in our code, there are 8 different analytical decision points in total, and we'll step through these branch by branch now. So the first decision point is whether you should adjust for covariates. And if you choose to adjust for covariates, this might be, with respect to large data sets, you might want to adjust for site or scanner type as a covariate. But you could also adjust for things like head motion, age, sex, etc. So if you choose to adjust for covariates, you have further decisions. You need to choose which sort of method you will use to adjust for covariates. So we enable 2 different ways of adjusting for covariates. So at the edge selection step, so when you are relating connectivity strength or edge strength in the connectivity matrices to your target variable, you can adjust for the covariates there using a partial correlation. Alternatively, you could adjust for covariates when fitting network strength to your target variable by including the covariates in the linear regression. You could also adjust for covariates at both the edge selection and model fitting step if you wished. The next choice you have to think about is what cross-validation method you should apply. So typically in CPM papers to date, leave one out cross-validation has been used. However, for large data sets, this will become very computationally expensive, and it might take an awful long time to run. So we'd recommend using k-fold cross-validation. This is also suggested, in some best practice guidelines for statistical reasons. But mainly it's more computationally efficient. And you can actually smooth out your model estimates or your predictions, your model estimates by repeating k-fold cross-validation a number of times. So your choices are what type of cross-validation to use. If you're using k-fold, the number for k, so usually 5 or 10-fold, so the number of folds. And then if you choose to repeat k-fold, the number of iterations. The next decision point is how you should select your features or select your edges. So typically the standard is to use a p-value threshold where p equals, where you only select edges that are correlated to the target variable with a p-value less than 0.01. Alternatively, some people have also tried to optimize these p-value thresholds by testing models with different p-values and then selecting the model with the highest R value. And this can result in overfitting to the internal validation data. So you should only really do this if you can test the model on external data and ensure that your model is not overfitting. Sparsity thresholds have also been used. For example, Abigail Green and colleagues in 2018 applied a range of different thresholds for only the top X% of the most strongly correlated edges were selected, ignoring their p-values. So Green and colleagues looked at different sparsity thresholds, including 1%, 2%, 5%, and 10% of the top most strongly correlated edges. So for visualizing and externally validating your model, you also need to choose the edge frequency threshold. So this refers to the number of folds or the number of folds in which that edge was significantly correlated with the target variable. So for leave one out cross validation, it's typical to use 100%. So you only visualize or you only externally validate with edges that were selected in every fold of the model. For repeated k-fold or just k-fold cross validation, this can sometimes result in a small number of edges. So you may be able to use less conservative thresholds if you wished. Again, if you're externally validating, this is fine. Your final decision point then relates to your permutation testing. Permutation testing is a way of assessing the statistical significance of model predictions on your internal validation or training data set. So it creates an empirical null distribution from the correlation between the predictive values from the model and the target variable that you can then use to obtain a permuted or randomly permuted p-value. So your decision here is to use or not use permutation testing. I'm sorry, the permuted p-value gives you basically tells you the proportion of random correlations that are greater than or equal to your true correlation value. So your choice is whether to use or not to use permutation testing, and if you use it, the number of iterations which you would wish to permute. So the standard in CPM has been to use 1000 iterations. But just to bear in mind, in the context of large neuroimaging data sets, this will become very computationally expensive when using leave one out cross-validation. So now we get to the example today. We'll be using the Amsterdam Open MRI collection dataset, and we'll be using task-based fMRI data from 861 participants there. This is also called the Ionic dataset. And so this is single site data, so we won't be using leave site out cross-validation. And just briefly, the data were pre-processed using the standard pre-processing pipeline. And then following that, 24 motion parameters were regressed out of the data. T-comp core was applied. A high-pass filter and spatial smoothing was applied to the data. And participants were only retained for analysis if they had mean frame height displacement of less than or equal to 0.5 millimeters. So we are trying to model the phenotype of fluid intelligence, and we would like to covariate for age and sex. So then, applying the decision points to our own example here, we will adjust for age and sex as covariates at the age selection step, so using a partial correlation. We will use k-fold cross-validation. Specifically, we will use 100 times repeated 10-fold cross-validation. And we will apply an edge selection threshold, a p-value threshold where p is 0.01. And we will only select edges for visualization and external validation that have been selected in 100% of the data, so in 100% of folds and iterations, so in all folds of the model. And we will run a permutation test with 1000 iterations of random permutation. So the data have been processed and are made available here very kindly by Emin Serene on his GitHub page. And there's 861 connectivity matrices here that are simply 268 by 268 connectivity matrices. And there's a design-IQ match file, which has, for 861 participants, our target variable of fluid intelligence, as well as our covariates of age and sex. So the target variable is in column 1, age in column 2, and sex in column 3. And just to bear in mind, 1 is equal to female and 0 is equal to male here, or denotes male. So if you follow the link here, you can download this data, and then you can download the code from the following GitHub repository. So flexible CPM, and just make sure to add all code to the MATLAB path, as this code base contains the functions and scripts that we'll be using today. So if you the relevant scripts I'll walk you through now are neuromethods prepare CSV CPM and neuromethods main analysis CPM. There are additional relevant scripts for the book chapter, which are leave site out CPM and computational lesion analysis CPM. We won't have time to cover these today, but they will be outlined in the upcoming book chapter. So the first step is to obviously download the data as instructed in Emin's link, and then open the following script. There's further information on downloading the data here as well. So this is the neuromethods prepare CSV CPM script. So all you have to do here is just make sure that the data is downloaded to the correct folder and you've correctly specified the directories here. So you will need to change this for your own machines. You then specify the different directories that we will use in the following steps. So you'll need to make sure these are relevant to your machine. And the script will then say output a connectivity matrix in the form of a CSV file for each participant in this folder here in the matrix directory. And it then splits the design IQ file into a covariates file and a target variable or behave data CSV file. Yeah. And this is not relevant today, but this, just for demonstration, the book chapter will make a sort of false site variable to pretend that this data set is a multi-site data set, so we can demonstrate leave set out validation. Again, this is not relevant today, so you can just ignore this bit. And you can even just delete this part of the code if you're not interested in following that example. So then you want to open up the main analysis script. And you will need to enter some further information here. So first of all, you want to check that the file paths are correct and are consistent with what was in the previous script for your machine. You then want to enter your target variable name. So here we've entered IQ. You then want to enter a name for your output file, so model name. So we call that IQ-CPM. And then you want to make sure that your number of participants is correct, so 861. Sorry, I'm just remembering. In the previous example, I by mistake left this as n equals 20 when I was just testing something. So this should be n equals 861. Just be careful with that. So you also want to specify your covariates. So if you are adjusting for covariates, you should have the name of the covariates here. These should be the same as listed in the CSV file, as should IQ, sorry. If you are not adjusting for covariates, you can just leave this as an empty string, so 2 quotes. And this script will then prepare the data and variables using the functions prep predictor CPM and prep targets, prep target CPM. So the next bit of the script, you will need to provide further model inputs. And this is where you'll specify the method for dealing with confounds or covariates. And so we have chosen relate here to relate the covariates to connectivity or to relate to covariates to the target variable. When we are relating connectivity to the target variable using a partial correlation. In the next bit, you'll specify your cross-validation scheme. So if you want to run leave without cross-validation, you just simply enter k equals to your n, so 861, for example, if you're running leave without cross-validation. But in our instance, we are running 10-fold cross-validation, so we enter k equals 10. You then specify the number of iterations you want to run. So we are running 100 iterations of 10-fold cross-validation. If you're running leave without cross-validation, iterations should be equal to 1. There's no benefit to running multiple iterations of leave without cross-validation. You then need to specify your feature selection type here, so thresh type. So we're using a p-value threshold, so we enter p-value. If you want to use sparsity, enter sparsity. And then you want to enter your threshold for feature selection or edge selection. So we are using a p-value threshold of 0.01, so that's entered here. You then need to also enter your frequency threshold for edge selection. So in our case, we're selecting edges from all folds. So we have a frequency threshold of 1 for 100%. We then also want to select whether you are running a permutation test. So we have said yes. Alternatively, say no if you don't want to run a permutation test. And then you need to enter finally the number of iterations of your permutation test. So we have entered perm iterations equals 1000 for running 1000 iterations of a permutation test. So after this point in the script, no further user input is required. So once you've got to this point, are you ensured everything is entered correctly? You can then just hit run. And the next section, it's pretty boring. It just pre-allocates arrays for computational efficiency using 2 functions, prep results arrays and prep parameters arrays. Nothing really interesting happening there. And then the next block of script is where the CPM is actually implemented. So using a parallel loop, so a par 4 loop. This section loops over the number of iterations. So in the case of leave one out cross-validation or a single iteration of k-fold cross-validation, it will just perform this block once. But in our case, we're running 100 iterations of 10-fold, it will perform this block 100 times. So the first section of this block actually executes the CPM run flexible CPM function. Then evaluate CPM is called to extract metrics of model performance, which include Pearson's correlation, R squared, and the mean absolute error of the predictions. And then the extract parameter CPM function is executed to extract the fitted model parameters from the linear regressions. And finally, the binary masks, which contain the selected edges in the positive and negative networks, are saved in each iteration. And then outside of the par 4 loop, the masks for each iteration are concatenated. And then following that, the extract edges CPM function obtains the indices or indices of the selected edges within the positive and negative networks. So finally, the last section of the script stores the model parameters, the predictions, the selected edges, and the results for model performance in individual structures. So these data enable reporting of the results, of the visualization of the results, and external validation of the results if you have an additional data set. And if multiple iterations of the CPM were conducted, then the parameters, predictions, and results are averaged across each iteration. So all this information is saved in a MAT file in the pre-specified output path with the pre-specified model name that we touched on at the start of this example. So when you load up, when you load up this saved file, you'll see the file it saves the following structures. So let's just look at model info, which basically saves everything all the model inputs, and you can use that to ensure that you're looking at the right model output. And then when you look at mean results, you'll see the average model performance across the 100 iterations of our 10-fold cross-validation CPM. So you'll see that we have a positive correlation between, or a correlation of 0.49 between the positive network strand predictions and the actual target variable. And the combined network is a bit better. It's an R of 0.177. So there is some predictive accuracy, although when you look at the R square values, they're actually quite small. So for the combined network, there's an R squared of 0.0315, so that equates to about 3.2% of the variance explained in fluid intelligence is explained by the combined network strength prediction. And then when you click on mean or perm results, this gives you the results from the permutation test. You can see despite the small amount of variance explained, the models are statistically significant. And then we look at mean parameters. These are the fitted model parameters for the linear regressions, and that you can use along with the edge indices for external validation. So saved here. And finally, mean predictions shows us the model predictions. If you click into each of these, you get the predicted predictions for each of your 3 networks and for each participant. So you also will have 2 text files saved in your output, and these contain the binary masks for your positive and negative networks. So to visualize these, you can open up Bioimage Suite Web. The URL is bioimagesuiteweb.github.io. So if you click on Applications Connectivity Viewer, make sure to first load your positive matrix. I think you will be prompted to always load the positive matrix first, but just do that first anyway. And you can then load your negative mask as well, or your negative matrix. And then on the right-hand side of the screen, in the connectivity control sort of toolbox, make sure that all is selected for mode. And 0 is selected for degree threshold so that all connections are visualized, and lines to draw is equal to both. So you show both positive and negative networks, which again essentially shows the combined network. This provides you with the following visualization, which shows the I think possibly the positive network in red, and the other color being the negative network. And you can mess around then with the connectivity control buttons to you know, only show the positive network, only show specific nodes, only visualize specific connections, and so forth. You can also export some of these plots too. So as I mentioned, there is additional functionality we don't have time to cover today, but the book chapter will demonstrate how to apply leaf-side out connectome-based predictive modeling for large neuroimaging data sets. I will also demonstrate how to apply computational lesions with CPN. So please keep updated with the book chapter, and you'll see how to do that. If you can't wait until then, the code is up on the GitHub, and it's well commented, so you might be able to follow along either. So finally, thank you all very much for listening and following along with this presentation. Of course, thank you to Herb Lamette and Rob Whelan for putting together the book chapter and organising this educational course. Thank you very much to Em and Sarin for preparing the aomic data for use. And importantly, thank you very much to Emily Finn, Jilin Shen, Dustin Scheinhost, and Todd Constable for making their work on the CPM open access and free for others to, like ourselves, to use, modify, and hopefully build on. And finally, thank you very much to other researchers within this Yale group who were very happy to help, assist, and respond to many different queries during the course of this work, including Corey, Steph, Genios, Abigail, and Mehrabeh. So thank you all very much, and please get in touch if you have any questions, and do download the code from GitHub, and hopefully you can make use of it yourself. So thank you. Bye-bye.